Yeah. So, good morning, everyone. Madungog ako, guys. Ma'am.
Yes, we can hear you.
Okay. So, good morning, everyone. So, today, uh, I will be discussing high throughput sequencing or HTS. So, in this discussion, I will be briefly introducing you guys to high throughput sequencing, as well as its importance, and also which will be followed by some of the more promising methods of high throughput sequencing, which are sequencing by synthesis, ion torrent sequencing, and nanopore sequencing. And this will eventually be followed by the applications of these methods in very recent studies. So first, what is high throughput sequencing? Now, a predecessor of high throughput sequencing is the Sanger sequencing method. Now, this was the method that was used to sequence the entire human genome 2 decades ago, 2 decades way back. Now, this method, the Sanger sequencing, had two prominent limitations. One was the low throughput, and two, the high cost. So, low throughput siya because every sequ- e- every pro- ano na, every run can only yielded several hundred base pairs, and relatively few runs could be completed in a day. So ang result is that low ra siya o output. Now, since this is a novel approach, it was also quite expensive. The cost was around $1 per base pair, and in a complete genome with billions of base pairs, ang pag-sequence niya would also cost billions as well. So, in order to compensate or to solve this problem, high throughput sequencing was eventually uh, born. This was through the modification of the Sanger sequencing technology. Some of the methods that I will discuss later on still employ fragments or some of the katong mga approaches na gigamit sa Sanger sequencing dayong gi-modify ra nila. So high throughput sequencing technologies are capable of sequencing multiple DNA molecules in parallel. That is to say nga simultaneous siya, which is millions ang occur in a single flow cell, then enabling hundreds of millions of DNA molecules to be sequenced at a time. So why is high throughput sequencing important? So important siya because HTS is used to create large data sets, generating more comprehensive insights into the cellular genomic and transcriptomic signatures of various diseases and developmental stages, as well as in sequencing the genomes of extant and extinct organisms, thus establishing evolutionary history. Now, the demand for understanding organisms is high today simply because there is a thrill when to establish a classification or to understand in general the entire biosphere. So, and the best way to understand this is through their genomes. Then HTS is able to meet this by providing the means to sequence genomes at a much more efficient manner. So, in here, as I said earlier, na ay three kabuok latest and most prominent sequencing technologies. One is sequencing by synthesis, second ion torrent sequencing, and the third is nanopore sequencing. Now, it is worthwhile to note that there are many more sequencing technologies out in the industry, but these three are few of the most promising. So sila akong gipili. The first is sequencing by synthesis. Sequencing by synthesis shears DNA into pieces and attaches PCR adapters in the 3' and 5' ends. And it uses bridge amplification method, which allows for the generation of small clusters with an identical sequence to be analyzed. And uses four fluorescently labeled nucleotides to sequence the tens of millions of clusters on the flow cell surface in parallel. Now, this is similar to the Sanger sequencing technology, in a manner that pareho silang gagamit of fluorescently labeled nucleotides, which emits light from the reaction when the nucleotide is incorporated into the strand. Now, this light, each nucleotide has a characteristic light or a characteristic wavelength na emit, and this can be interpreted by a computer to represent a base call. However, ang difference niya with regards to Sanger sequencing is that the dyes na gigamit ani the dyed nucleotides na gigamit sa sequencing by synthesis do not terminate the elongation as Sanger sequencing does. So Sanger sequencing man, once a nucleotide is incorporated into the strand, muhunong na siya o terminate. And so the result is that line o lengths, sequence lengths ang ma-generate sa Sanger sequencing. So here is a brief process. So first is DNA, the isolated DNA is fragmented into smaller input-sized fragments. So the process of fragmenting can be done in quite a number of ways, but as I have researched, one of the most efficient is through sonification, which is through the use of sound, ultrasound actually. So to break the DNA into fragments. Now this fragmentation is not a result to an uneven fragment. So as you can see, dili siya even there are blunt, dili siya dili gatupong ang iyahang end sa mga fragments. And so there is a need to repair. So in this case, the missing pairs are recovered, are regenerated so as to produce this even ends para blunt na siya. And after that, adapters are attached or ligated to the 3' and 5' ends. Now, the adapters are essential because they are integral sa process ng sunod, which is the flow cell hybridization. So, in this case, ang flow cell hybridization, katong mga fragments is mo hybridize siya sa flow cell. The flow cell is appeal na siya sa setup aning method, and on one end of the adapter, one end of the strands or the fragments attaches to the flow cell, and then mo bend siya so that the other end would also hybridize. Then the next step would be to generate multiple clusters and individual strands. Now this is important because this ensures nga makita siya, ma-detect ang mga strands or later on kanina nga step, ma-detect siya by the computer, and this also assures nga ang accuracy niya is taas. Then the reverse strand is removed because ang forward strand na may gamiton. Then the nucleotides would be attached aning mga forward strands. Then again, similar siya sa Sanger sequencing, once those nucleotides are incorporated into the strand, they release a characteristic light, which can then be interpreted by a computer to signify or to represent a base call. So in this case, yellow, when this mystery nucleotide is incorporated, it releases the yellow color. The computer then uses the algorithm that it has been encoded into, that has been programmed into, and interprets that yellow light as the base T. So, in effect, since ang iyahang sequence, ang iyahang na-detect is T, therefore, ang iyahang pair is A. Then, repeat na siya ani nga sa second cycle. So, in this case, ang na-encode niya o ang na-detect niya nga light is green. So since green siya, i-interpret sa computer, then that means A. And since A siya, then that means ang iyahang pair kay T. Dayon, afterwards, since remember sa first step niya, the DNA was fragmented. So, gagmugbo ra jud na siya nga strand. So the next step is to align the reads nga katong ginagmay ng mga strands to produce the unassembled sequence. Next, so ang advantage aning sequencing by synthesis is its potential for high sequence yield. Although depending na siya sa sequencer model o sa desired application. Now this is true because iyang putlo ng DNA and create siya og mga clusters. So ang iyang mahitabo is that you could repeat a you could repeat the se- the s- na, the sequences the sequencing to produce uh, many copies. That would make your research the process accurate. For instance, it is estimated, it has been quite proven nga for maka-generate siya og ang a single sync process for 10 days, worth 10 days na sequencing with synthesis can generate up to 6 gigabytes of data. And that 60 gigabytes is worth 30 kabuok copies anang imuhang genome nga sequence. So high jud siya og throughput and at the same time accurate po siya because dahan man copies. Disadvantages on this method, however, is that equipment can be very expensive. A single machine developed by Illumina, which is the pioneer aning method, cost around $650,000. So mahal jud siya. And another is that it cannot also sequence long reads. Kay mugbo raman iyang kaya. Next, the next method that I will discuss is ion torrent sequencing. In many ways, ion torrent sequencing is similar uh, to the sequencing by synthesis method, wherein ang DNA is a fragment code. Yeah. However, in this case, dili siya mogamit to flow cell. Ang iyang gamiton is microscopic beads, wherein DNA fragments attaches into kani siyang nakita dire. Then, a difference with the sequencing by synthesis is that it does not use light and instead it uses the release of hydrogen ions, the detection of released hydrogen ions to signify a base call. So I will discuss that you. So first is that DNA is captured onto beads as shown here. Matapot siya sa beads. Dayon, this single DNA is amplified. Sumudaghan siya through PCR. Then, kana siya is i-contain na siya into a well, a micro well. Now, this is also is inside or is in a plate. Pareha atong kaniha mag flow cell plate but naa siya mga well sa lalong wherein the beads can be contained para dito mo undergo ng sequencing. Then, once this happens, muna yung mga nucleotides nga gi-flow pasulod sa cell. Then, once naa siya mo incorporate siya og nucleotide sa sequence, mo release siya og proton which can be detected by the sensor na nas lalong sa well. And then, since na siya na-detect na hydrogen during a particular flow of nucleotide, and that would mean na kung unsa man tong nucleotide nga gipasulod, type of nucleotide nga gipasulod would signify kung unsa na incorporate. So for instance, if in one time ni flow siya og thymine nga nucleotide, then nipagawas o nakadetect og hydrogen or proton, nakadetect og proton, that would mean nga na-incorporate sa sequence tong thymine, which would also mean nga A or adenine ang naa sa sequence since T man iyahang pair. Ra? Dayon, so mo to siya iyahang process, detection of hydrogen ions. Now, advantages of this method is that less expensive ang iyahang equipment, or at least least expensive compared to the first method, but still quite expensive, around $350,000. And it is also fast. Ano yung advantages niya? A disadvantage, however, is homopolymer errors, which are errors in the number of bases called when a single nucleotide occurs more than once in sequence. This is important to note because ang detection manggod niya is by the number of hydrogen or by the release of hydrogen ion. So if magsunod siya nga puro siya, in this case puro siya G, the hydrogen ions nga ma-detect could be interpreted as a single base call. So instead of 3, ma-record siya as 1, which is already an error. So next method is nanopore sequencing. Nanopore sequencing is a very novel method. If I'm not wrong, it was launched 2 years ago, I think. And this involves through by monitoring changes to an electrical current as nucleic acids are passed through a protein nanopore. The resulting signal is decoded to provide the specific DNA or RNA sequence. So, Now, this is a protein nanopore and was inspired by the nanopores that exist on the plasma membranes of cells. Yang himon is that this uh, enzyme unwinds the DNA, and one of the strands goes into the pore, and as it does, moagi siya ani na electrical current diri nga part kaning sa membrane, which upon upon the crossing of a strand is disrupted, and this disruption can be interpreted by a computer to signify a base call. So for instance, if yang sequence kay ang yang nucleotide kay adenine, then that would be nga ang disruption niya is specific to this specific sa usa ka current nga recognizable by computer as seen aning gamatoy diri nga figure. Advantages aning method is that it has the longest individual reads. As you may have noticed, wala siya ni undergo og fragmentation, meaning that wala gini lagi putol-putol tong DNA, then gidaritso ra nila og pansak sa gidaritso ra niya og pansak sa nanopore, gidaritso ra og basa. And it is also portable. The recent devices na available aning nanopore sequencing, which was developed by Oxford, by the way, is only the size of a smartphone. So, and the fun fact is that this device can actually be carried around and be plugged into a portable computer, such as a laptop. So, pwede siya magamit ball. So, uh, if na siya kailangan siya og complete na laboratory in order to be accomplished. On the contrary, kaning nanopore sequencing, which does not fragment DNA, has problems with accuracy, although gamay ra pod. Nandili siya ma-validate. However, it is efficient in a sense that pwede siyang magamit when it comes to field studies na lang nara ka sa field. So it depends on what particular factor of efficiency you're looking into if mopili ka aning duha.
Accuracy, fragmentation is more restricted than simplified. Okay. How about the time spent?
Okay ka, ma'am.
Choppy ko, choppy.
Okay na, ma'am. Hello?
Oh, can you hear me?
Yes, ma'am.
Anak,
Okay na.
madungog mo ko. Oh,
Yes, ma'am.
instead for fencing. Sa mas dugang. Planted nga, best or this method?
Which is better, ang fragmented or this, ma'am?
The time spent, asay mas dugay? The fragmented method or this one?
Ay, kuan siya, ma'am. Actually, pareho ra siya, ma'am, but mumatter ra siya, depende if unsa kataas ang sequence o unsa kataas ang strand nga gi-sequence aning sa nanopore, because binagsaon man niya, whereas when it comes to high throughput sequencing, pwede man siyang pwede sa iyang the manner nga iyang putol-putulon means makasequence siya og an entire genome actually in a single day. But if mubo-mubo ra pud ang sequence, pwede ra pud siyang ma-apply diri sa nanopore.
Which means that it can sequence DNA molecule simultaneously or DNA simultaneously. That's for the case of first two methods, right?
Yes.
For this one, this just DNA molecule has weight before it can sequence the whole thing. Okay.
Yes.
Do you have napanisip mo pa yung Mr. Montegrejo?
Napay disadvantages, ma'am, then related studies pa.
Okay. Go.
Okay.
I just want you to, after you discuss the disadvantages, can you please tell us what is its difference with NGS, and then the microarray, if you have.
Okay, ma'am, sige. So, disadvantage of this method is lower siya og throughput since suggested, as indicated, magbinagsa siya og long read. So there seems to be a conundrum wherein the longer read a sequence, the lower the throughput that is yield. Although this is only so far as our technology is capable of. Then also, the error rate can be up to 5 to 10%. So 5 to 10%. So mas taas siya og error rate compared to the other methods. And the reason for that is because little siya method for validation, because unlike the previous methods, dahan man kang copy, so there is room for validation afterwards. So in this case, wala man, because dili man niya gi-amplify ang DNA nga imuhang makuha, diretso ra man niya, so wala room for validation. So mauna siya. Then next is the related studies. So this study that I found that was published on the 7th of April 2021 by Haj Jinja, and was titled The Initial Upper Paleolithic Humans in Europe at Recent Neanderthals. So, ang title is
Hello,
Initial
is
Paleolithic
it-
Humans in Europe had Recent Neanderthal
Hello,
Ancestry.
is
So,
the
in this
internet
study,
connection of yours,
they use
Mr.
Illumina
Montefiore?
sequencing, that is sequencing by oh dear, wala ni Bimo. Okay na. Hindi mo
Is it my internet connection? Hindi ko. Or gaputol-putol ka. Basik ako ni. Sige, go on, Mr. Montegrejo.
Okay. So, in this study, they used Illumina sequencing or sequencing by synthesis to sequence the genome of 3 individuals. Hala, ako daw the internet. Meron ko. Okay. Asan ako na to? So, ang ilang gihimo is that they sequenced the genome of 3 individuals, which was dated to have lived between 45,930 and 42,580 years ago from the Batshokiro cave in Bulgaria. Now, in this study, they found out that these individuals are closely related to present-day and ancient populations in East Asia and the Americas, and that these individuals had Neanderthal ancestors a few generations back in their family history, thus confirming that the first European modern humans mixed with Neanderthals. Now, there was speculation that Neanderthals and modern day humans actually interbred in the past. So this study simply confirms or adds evidence na indeed there was interbreeding between the two because as apparent, since these individuals were related to modern day East Asians and Americans and that they had Neanderthal ancestors way back, uh, yes, in a few generations back in their family history, suggests that the Neanderthal DNA therefore resides in modern day humans. And it was all possible because of Illumina sequencing or the sequencing by synthesis method. Next, solution change. Why, why? Okay. Now now another article, uh, which was published on the 13th of January 2021 by Angela Perry and others, entitled Direwolves Were the Last of an Ancient New World Canid Lineage. Now, for those of us na fans sa Game of Thrones, this familiar siguro ta og mga dire wolves and familiar sa dire wolves and for ages ever since their discovery in the 19th century, dire wolves were classified under the genus Canis, which also includes modern day dogs, wolves, coyotes. So in this study, they sort of debunked that particular aspect when they discovered through the use of Illumina sequencing or sequencing by synthesis that direwolves are in fact highly divergent to modern day canids and was actually and did actually split from that lineage about 5.7 million years ago. Now, this is important because gray wolves, which were once closely considered, which were once considered as the closest relatives of dire wolves, were very similar in morphology to dire wolves. And but however, after this research, after this study, na-confirm nila nga dili di ay siya closely related to any of the members of modern day of the genus Canis. So, ang nahitabo is that muratag nauwat sa iyahang morphology sa ilahang similarity sa morphology. Now, this is important because we classify organisms today through morphology. And oftentimes, when they are very much alike, we designate them the same classification, in this case, the same genus. So by enabling or by viewing their genomes through sequencing technologies, maka-verify ta whether our classifications via morphology is worthy of merit. So, importante siya because it can give us an overview of evolution. Ano. So, muna.
Okay, can you discuss its advantage or disadvantage over the previous methods that were discussed by your classmates or by the reporters?
Yes. So, wala ko slide anam mo kaya.
Sa inyong advantage or disadvantage of techniques?
Yes. So with regards to NGS, actually, high throughput sequencing is actually subsumes NGS because katong akong gidiscuss nga first two methods are actually next generation sequencing technologies. However, this differs, high throughput sequencing in general differs with NGS because it is open for other types of sequencing methods, particularly katong akong gidiscuss nga nanopore sequencing, which is already a third generation sequencing technology na mo far exceeds sa NGS. So, in essence, kung unsa tong mga gikulangan sa NGS are provided by some methods of high throughput sequencing. One of these problems is katong ilahang the inability of NGS to read or to sequence long reads kay as you might remember, putol-putulon man yan ng DNA. So in the case of high throughput sequencing technology, particularly those of the third generation sequencing technologies, there is no need to cut the DNA into fragments and pwede na siyang gidiretso og feed sa uh, sa machine na gamiton. Disadvantage, however, is that although this might be the case, there are issues with regards to the level of accuracy of third generation sequencing technologies because there is very little room allotted for validation of the data since there is only one copy to begin with, unlike with NGS which amplifies the amount of data that is present so that validation can be conducted later on. Kay ang error when it comes to sequencing is still significant, and so is still significant in both kinds of technologies. But ma-overcome na siya with NGS because they are able to make more copies, which is not which is an incapability when it comes to third generation sequencing such as nanopore sequencing. Yeah.
Okay, the method HDS, also known as third generation sequencing, is an improvement to the previous method NGS.
Yes.
Okay. And as for the microarray, what is the advantage?
Ah, ang advantage niya, ma'am, is that when it kuan ito ba advantage niya? Ang as far as I have understood with last meeting, ang microarray is almost exclusive to gene expression, if I'm not wrong. And so ang limitation niya is that if there are certain uh, conditions wherein a particular gene is not expressed, then dili siya mabasa. So that is overcome uh, by HTS because there is no need to rely on gene expression because ang whole genome may gi-sequence niya. So pwede na siya diretso basa
Okay, do you agree with that, Mr. Banabudos? Mr. Banabudos? Hello? Is it exclusive to gene expression?
Dili, almost exclusive. Almost exclusive.
Okay. All right. Although it also deals with SNPs. But this one also deals with SNPs, right, Mr. Montegreo? Okay, naman
Yes.
siya evolutionary.
Yes,
Okay,
ma'am. There
but
are,
yeah, as far as yeah, as far as I have read so far for articles that deals with this type of researches like evolutionary history, taxonomy, molecular marker, use molecular morphological marker,
happy ka, ma'am.
Choppy ka, ma'am.
yes. Hello? Chaco?
Happy ka.
Sorry. I'm sorry. Shopping ako?
Yes, ma'am.
Oh, may globe. Oh,
Lumipat ka na sa smart.
hello? Can you hear me?
Yes, ma'am.
Hello?
Mawala ka, ma'am.
Sana nalimot sa ingon. Nakisra man buhay mo. Kan putol-putol gapon siya gapon.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay. Let's move on to the next repeater na lang. Mr. Migo, let's move on to Mr. Nas before Whisper.
Mayo na, guys. Makita na? Something. Okay, go. So, good morning, everyone. So, before last week, I already discussed about genotyping. What is it? And its advantages and disadvantages, and its importance. So now, I'm going to discuss some of its related studies. Um, in with respect to its importance in the four fields I've discussed last week. So in agriculture, um, high throughput SNP discovery and genotyping in hexaploid wheat. So in this study, it reports about on the use of whole genome resequencing data from 8 wheat lines to mine for SNPs in the genic, the repetitive and non-repetitive intergenic fractions sa wheat genome. And so like because of um, its abundance and its amenability to high throughput genotyping techniques, such as the single nucleotide polymorphisms or the SNPs. It is a powerful tool for efficient applied research. So going to forensics, the study of Jingzhu et al. The genotyping polymorphic microhaplotype markers through the Illumina microsequencing platform for forensics. So this study, the microhaplotype markers were evaluated if it could really be a new choice in providing forensic practices in the future. So by the way, ang microhaplotype loci or the microHAPs, or also called MHS, are a novel type of molecular marker of less than 300 nucleotides, which has a really close link of SNPs. Yeah, really close links of SNPs. So these haplotype markers have received broad attention in forensics, and because of its capability to supplement existing genetic marker panels. So the present genetic markers we have today includes the strips and the SNPs. Strips for short tandem repeat polymorphisms, and SNPs for single nucleotide polymorphisms. But these two have drawbacks. The stutter and the high mutation state of STR markers and the low polymorphism of SNP markers, which can obstruct the solving of certain cases, which solving of certain cases. So in this study, uh, they they have so KID proposed microhaplotype markers that encompasses 2 to 4 SNPs. So in this study, they used the microhaplotype loci into they were I'm sorry. In this study, microhaplotype loci were screened through 3 criteria, and they chose the Illumina mic sequencing platform to sequence the these new markers. And so they found out that um, the microhaplotype markers um, could be used for individual identification and ancestry inference, and it is a new choice for providing forensic practices in the future. So the next study is in medicine, which is the genomic association study of severe COVID-19 with respiratory failure. So this study by the genomic association analysis allows for the identification of potential genetic factors that were involved in the development of COVID-19. So since there has been a considerable variation sa disease behavior among patients that are infected with SARS-CoV-2, this 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 study is significant. So the results of these studies detected cross-replicating associations with the RS11385492 at 3P21.31 and with RS657152 at locus 9 Q34.2. So they found or detected a cross replications between these two locus, which is significant at the genome wide level. Also, they found out that at the locus 9Q34.2, they found the association signal coincided with the blood ABO group locus, which means this unit, a blood group specific analysis showed a high risk in blood group A than in other blood group. So meaning naay ang blood ABO groups shows higher risk to COVID-19 than other blood groups, specifically the blood group A. And they've also found out a protective effect in blood groups O as compared with the other blood groups. So, in conclusion, they identified that 3P21.31 gene cluster um, gene cluster as a genetic susceptibility locus in patients with COVID-19 with, with respiratory failure and confirmed a potential involvement of the ABO blood group system. So next study, basic scientific research by Chang Huan Yin entitled Genotyping Coronavirus SARS-CoV-2 Methods and um, Implications. So this study was conducted to understand the evolution and transmission of SARS-CoV-2 by genotyping of virus isolates, genotyping of virus isolate. So this study presents an accurate method for effectively genotyping SARS-CoV-2 viruses using complete genomes. Um, um, its method employs multiple sequence alignments of the genome isolates with the SARS-CoV-2 reference genome. So, and then ang iyahang SNP genotypes are then measured by Jacquard distances to track the relationship of the virus isolates. And so, ang iyang results, the genotyping analysis of SARS-CoV-2 isolates from the globe reveals that specific multiple mutations are predominated mutation type during the epidemic. So, kani siya nga proposed method served an effective tool for monitoring and tracking the epidemic of pathogenic viruses in their global and local genetic variation. So this genotyping analysis also shows that ang genes encoding the S proteins and the RNA polymerase, as well as the RNA primase and nucleoprotein, undergo frequent mutations. And lastly, these mutations are critical for vaccine development and disease control. Ano naman yung last study? Marathon. That's all. Ilagay ko kadungog ninyo.
Naramitin.
Yes, ma'am. Tessa, ma'am. Makita na, guys.
Yes, Marg.
Okay. Okay, good morning, everyone. So, good morning. Today, I'll be discussing about CRISPR-Cas9, a tool emerging in genetic editing. And I guess some of you are already familiar with this. So let's start first with the outline on the introduction of CRISPR and how does this tool work and the applications and also the relevant studies apply regarding about the applications of CRISPR gene editing and its pros and cons or its advantages and disadvantages. So what is CRISPR? So bacteria and archaea are also prone to being infected, and these microorganisms have also evolved a defense mechanism to fight viral infection, particularly bacteriophages or phages. Now, CRISPR, as its name implies, clustered regularly interspaced short palindromic repeats, Cas9 or CRISPR-associated proteins 9 are molecular scissors that can be programmed to search for, bind, and cut only specific DNA sequence. So this has its superior end because it has its specificity in compare with other gene editing mechanisms. So when a bacterium encounters a virus for the first time, it stores the viral genome to its bacterial chromosome or its genome as a more like a genetic vaccination card for them. So cutting the viral, incorporating the viral genome can help them to cut viruses DNA and messes up with its function and also disrupts the life cycle of the virus. And this mechanism also allows organisms to, record over time, katong gingon mo siya genetic vaccination card. So it serves as a adaptive immunity response for these microorganisms such as bacteria and archaea. So CRISPR has a associated protein which common is Cas9. This set of genes allows for the production of different Cas proteins that has the ability to cut certain sequences of DNA on the virus. And also, a the guide, can I explain? This CRISPR locus or CRISPR array is composed of repeats and spacers. And these repeats are obviously repetitive sequences that vary about 23 to 55 nucleotides and is highly conserved among species. And also, this genetic material or sequence flanked by the repeats are called spacers, which came from the virus or derived from the virus's DNA or genetic material. And this serves as their vaccination card for incoming similar viruses with the same genetic material. So next is I'm going to explain how CRISPR works in bacteria or microorganisms. So first, there are three different mechanistic steps in this adaptive immunity response of bacteria or archaea. Now, first is acquisition. Acquisition is where this bacteria acquires the genetic material of the virus or the bacteriophage from its genome to further um, to further um, operon is the transactivating CRISPR RNA or tracer RNA here, the orange rectangle, which encodes a unique non-coding RNA with homology to the repeat sequences. So this tracer RNA is homologous to the repeat sequences of the CRISPR array. Now, in acquiring the viral genome, the next step would be expression of the Cas genes and producing those RNA to infect to in DNA interference. So in expression, in expressing the Cas genes and also the tracer RNA gene and also the spacers, the spacers are also co-transcribed with all other spacers into a long precursor CRISPR RNA or pre-cRNA. So we have here the pre-cRNA composed of the proteins transcribed from the tracer RNA gene, the Cas genes, and also from the spacers or in the CRISPR array. So in transcribing, the tracer RNA is transcribed separately and then recombines with the pre-cRNA. Here, the pre-cRNA and tracer RNA are in different transcription process. So in pre-cRNA and tracer RNA, they are undergoing maturation by a specific enzyme called RNase 3, and also there is trimming occurring. So when the pre-cRNA is matured, cRNA will be produced, and this cRNA will bind to the Cas protein. Here, as for this case, is Cas9. Cas9 came from this Cas operon, this specific gene. And this different kinds of gene has also the ability to function in acquisition machinery. So in DNA interference, the mature cRNA or CRISPR RNA with the tracer RNA, here we have the tracer RNA and the mature cRNA. This complex is now called guide RNA, which has the ability to bind with a specific sequence when a similar virus or phage comes to infect the bacterium or archaeum again. So when in interference, the binding is helped with the PUM or protospacer adjacent motif, which has the ability to further specifies on where the guide RNA and the Cas9 protein would bind to. Now, it has the ability to bind to the genome of the phage that would infect the bacterium, and also to cut it out and further iyang i-cut, and then ang function sa virus will never be infecting the bacterium or archaea again. So those are the 3 mechanistic steps involving CRISPR in the adaptive immunity response in bacterium. Next is the mechanism of CRISPR-Cas9 in genome engineering. Now, it is somehow similar in the mechanism of CRISPR-Cas system or complex in eukaryotic organism. So the mechanism of CRISPR-Cas9 mediated genome engineering is that they have a single RNA, which is composed of CRISPR RNA and tracer RNA, which was discussed prior this. And also, it's a pre-designed guide RNA to further bind to specific DNA sequence to on what on the specific genome of the desired on what we desire to cut out on the genome of eukaryotic organisms. So in the in this process, the the CRISPR-Cas9 complex will cut out the specific genome, the specific sequence of the DNA with the help of PAM. And also, when they cut that out, they produce double-stranded breaks, which would either undergo NHEJ or non-homologous end joining, which can produce insertions or deletions or indels, which may disrupt the function of the gene or may produce other effects on the organism. So in other way, in the presence of a donor DNA or donor template containing the sequence of interest, so HDR comes into place. HDR or homology directed repair can be initiated to create desired mutations unlike NHEJ. And this type of mechanism can produce our desired mutations, our desired sequences on what will this particular sequence of the DNA would contain. Next. Next is the application of CRISPR-Cas9 in different fields of biology and its relevant studies regarding its application. So in agriculture, this tool is also implemented or used in conferring disease resistance in plants or crops for acquiring desired traits also. Now, in this study, CRISPR-Cas9 was used to improve the strength and the lifetime of the crops. And since traditional or conventional methods are indeed consuming and labor intensive, in the researchers just here in the Philippines developed a disease-resistant crop that would help in the improvement of agriculture. That's in 2018. So here we have different, the Oryza sativa or rice have been affected by rice tongue root disease caused by a virus, which is RTSV. And in their study, the CRISPR-Cas9 system was used to mutate this specific gene named this specific gene, ELF4G gene, in Oryza sativa and develop RTSV resistant varieties that could potentially be released to the market that would be resistant to the virus. And also, the induced mutation were successfully transmitted to the next generation, which is a good thing. So in medicine and research, there are various applications of CRISPR-Cas9. Now, CRISPR-Cas9 is widely used in fixing genetic diseases like sickle cell anemia, beta thalassemia, and other genetic diseases. But here I'll be explaining what CRISPR-Cas9 would do or have the ability to become a diagnostic tool for COVID-19. In this study, they developed a CRISPR-based diagnostic assay which has the ability to detect the virus, specifically the the SARS-CoV-2, even after it has gone through mutations, which is their edge on different diagnostic tools regarding CRISPR-Cas9. There are a lot of diagnostic tools that use CRISPR-Cas9, but this has its edge. And also, it also has the ability to diagnose the disease without the need of RNA purification, and that would lead to faster rate of production of results. This 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 group of researchers are from Singapore, and they produce this specific test called the Vanguard or variant nucleotide guard. So how does this specific test detect the disease even after mutations? It has the ability to uh, detect the virus even if has undergone through mutations, because they use um, two guide RNAs that could recognize sequences that are extremely similar with different variants of SARS-CoV-2, since there are different variants of SARS-CoV-2, and also that are unique to the virus itself. And each guide RNA um, um, was presented to be 99.5% um, of that, similar to the thousands of SARS-CoV-2 isolates from their study. So combining the two or more guide RNAs with the enzyme, they have a specific Cas protein that they used here. They have Cas12 and not Cas9. So in this specific type of Cas protein, they have developed two or more guide RNAs to bind with this Cas protein, and then that would ensure that if one of the guide RNAs fails to guide it to the correct segment of the virus, of the viral genome, because of a mutation, the guide RNA can still rescue this mismatch. So they have a preserved nga guide RNA to detect if ang usa ka guide RNA could have a mismatch, which is very relevant as to this day and age. So they this is a very easy diagnostic test to deal with. They are composed of just paper strips dipped into a tube containing the crude samples and the reaction mix. If one band, that would represent negative. If two bands, that would represent positive. Next would be the advantages and disadvantages of CRISPR-Cas9 genome editing. So the advantages of CRISPR-Cas9 is obviously to be cost-effective, just like the study a while ago where they used CRISPR-Cas to be a diagnostic tool for the emerging disease COVID-19. And it can also be used into even if a person has a limited experience on this field. And it also can be used in a wide array of organisms, unlike other genome editing tools. And also, the disadvantages of this tool in genome editing, they can have off targets, especially the NHEJ mechanism of in a host mediated repair mechanism. And double-stranded breaks are also error prone. And also, in today's age, the era of genome editing raises a lot of ethical questions that would probably that would need to be addressed by scientists and the society. So I have here an example of how CRISPR-Cas9 genome editing would affect the society if kanang pasagad lang o gamit without hesitation. So this particular Chinese biophysicist named Zhuangqi, he used CRISPR-Cas9 to create a 32 base pair deletion in the CCR5 gene. CCR5 gene is a genetic variant that confers increase in resistance to HIV infection. If iyang create ang 32 base pair deletion in CCR5 gene that would confer, ang iyaha is ma-resist ang duha ka twins nga iyang experiment in the HIV infection, since ang father sa duha ka ang father sa twins is HIV positive. So he used carelessly the CRISPR-Cas9 to create a 32 base pair deletion in the CCR5 gene. Now that's his goal, but the outcome became devastating, since we have no idea on what would be the outcome of this CRISPR-Cas9 genome editing. So he worked in secret without knowledge of his university, and also he just uploaded a video in YouTube on how he come up with this and how he performed this experiment. Now, the modification of the human germ cells, since he used human germ cells in this one, and the result was indeed devastating, and the result was this one. Ang iyang goal is 32 base pair deletion in this specific gene. And then ang na-acquire sa twins is very much different to its goal. So si Lulu and Nana, which are twins, they develop a 15 base pair deletion and a 4 base pair deletion in Nana. So the results are not similar to his goal, and he was indeed drew a crowd of journalists and bioethicists whom have been debating on the risk of gene-edited humans for years, since we have no idea on how CRISPR-Cas9 would work in human germ cells. Next, we have Victoria Gray, a survivor of sickle cell anemia and the first person to get treated in the United States with the technique CRISPR-Cas9. Now, in this in this situation, Victoria Gray indeed developed the desired mutation, and she has also developed an advantage in using CRISPR-Cas9 since she hasn't required any emergency room treatments and 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 hospitalizations or blood transfusions after the treatment, which is a very good thing for CRISPR-Cas9 in its emergence in genome editing. So I have also a study regarding CRISPR-Cas system, but it's way different from the usual CRISPR-Cas since this study included CRISPR-Cas9 to be highly modified to somehow search and then have specific alteration the in genetic mutations, like in the case of progeria. Well, here, CRISPR-Cas was modified into being not able to cut specific DNA sequence, but it has the ability to bind and also to have a base editing, which replaces the specific genetic mutation, like in this case in progeria, and um, single nucleotide ra iyahang kwaon, which is very much different to CRISPR-Cas9 since the usual CRISPR-Cas9 is uses guide RNA, which acquires a long sequence of um, DNA to cut it out. But this one, just a single nucleotide. So here he have developed base editors that would transform or replace specific nucleotide, and also they this ability or this tool, this mechanism has the ability to minimize those undesired editing, which is very much common to the usual CRISPR-Cas9. So in the usual CRISPR-Cas9, it is somewhat molecular scissors, but this one, base editing or prime editing, this is more like a pencil that is capable to directly rewriting the DNA letter into another nucleotide. Yes. Okay. Tessa, wala ko kabasa. What is the function of the yes? The function of the endonuclease, specifically the Cas proteins, they have the ability to become molecular scissors or cut out specific DNA sequence in a genome or in a genetic material of different organisms. How does it differ with Cas9 in terms of function? Cas9 sa unsa man katong different Cas proteins like Cas12 12 and CAS 13. Check to ba? Okay, ako na naman answer. In the case of here, kato akong gipakita nga diagnostic tool sa researchers sa Singapore, they use CAS-12 in which is very much different to CAS-9. Asa to siya? Here. Which is very much different to CAS-9 because CAS-9 has two different nucleus domains, which are HNH and RUVC, and they have, and this kind of Cas protein has the ability to bind to RNA, which is the genetic material of SARS-CoV-2, which is single-stranded RNA, right? How is the application in production? Oh. Well, CRISPR-Cas9 has the ability to edit a particular sequence of a genetic material. So it has the ability to probably to detect different mutations that varies from person to person, or SNP. Yes, some fragments are indeed conserved in species, specifically on the repeats. They are highly conserved, especially if species are in the same one species. Thank you, ma'am. Storya, ma'am.
Can you hear me? Okay na? Internet?
Yes,
Hello?
ma'am. Yes, ma'am.
Okay. So, Mr. Salapa, wala lagi Salapa, to report the article that I last time. Well, online this coming Thursday, no? Friday. Hello?
Choppy na po.
Thursday nga.
Choppy.
Chop it.
Thursday.
Okay, ma'am.